Hello and welcome. On today's program, we're going to be speaking with Dr. Marius Bauza. He is one of the doctors at the University College of London and part of the group that was winner of the Nobel in 2014. Fundamental to being alive is to know where you are, where other things are, and the relationships between them. Those distances and directions are essential to maintaining life. Deep in our brain, there's a small structure that the early anatomists thought looked something like a seahorse, and so they named it the hippocampus. Unsure what it did, the simple structure that appears in humans and many mammals was a mystery. In the early 20th century, the great psychologist Tolman deduced that there had to be a central location in the brain that would create a cognitive map, an understanding of place and environment and how we fit into it. The interactions and concepts, he thought, had to be present in one location. In the Milner studies, years later, an epileptic patient, simply known as HM, who had surgically lost his hippocampus in an effort to subdue his epilepsy, became a watershed instance to clarifying and understanding that the hippocampus dealt with memory in a profound way, and specifically spatial memory, and was dedicated to memorizing spatial events. Fast forward a few decades, and the meticulous and painstaking study of doctors like John O'Keefe would unveil an important facet of this little portion of the brain as he discovers in the early 1970s. He discovered place cells, a single cell, a single neuron that lights up and identifies when you're in a specific place. One cell is connected to one part of the room, take a step, and a different cell lights up. It seems like Dr. O'Keefe found a fundamental navigation system in the brain in the hippocampus. Years later, grid cells would be discovered by May Britt and Edward Moser. These cells act when if you're walking around an environment, they don't just respond in one place, they respond in a whole bunch of places, but in a pattern. And that pattern adds up to a grid, which can allow greater freedom in navigation and in improvising to reach a destination. This is a different and complementary navigational method that operates in that same portion of the brain, the hippocampus. Today, we discuss the continued progress of studying the hippocampus and the GPS of the brain with Dr. Marius Bauza from the Nobel-winning team at the University College of London, and specifically, we get to talk with him about his work on boundary cells. We start out with a brief discussion on how he got into the study of the hippocampus. This is a portion of the conversation with Dr. Bauza. So what was your focus when it came to physics?
So in physics, I was working on solid state physics. So I was looking at detectors, radiation detectors. It was really hardcore physics, material physics. And afterwards, in engineering, it was too. It was quite material orientated, but it was also electronics. And because of that, my profile was perfect for literally working with silicon probes. So, at the moment, that's what I'm doing. So all these things you see are, in a way, side project, but very, of course, fortunate side project. But my main project is working on devices to record from the brain.
Wow. I think that's one thing that I kind of got out of the study of the subject so far, is that when we take a look literally from Tolman in the 1930s and 40s and his whole idea of coming up with an idea of cognitive maps, that there has to be some central place in the brain where this is all happening. And he, operating on a hunch, he was right. But it really takes until we get to PET scans and fMRI before we could really do anything about it, in some ways.
Yeah, so it depends how you look at it. So I think the person who proved that or at least moved the whole cognitive map understanding further was John O'Keefe. So because as you said, when Tolman proposed the cognitive map, it was in a way that there must be some structure where we store this mental representations, where we update them, use them to understand where we are in the environment. But what he did, what John O'Keefe did, he showed that actually in hippocampus we do have cells which we would expect to have in the brain area which is working with cognitive maps. So it was namely play cells. So he observed that some of the cells are active only in some part of environment, not the other, and there are multiple cells like this. And that's how he started forming this hippocampus as a cognitive map theory.
Mm-hmm. You were part of the team when John O'Keefe's team when everything kind of came about, and congratulations is due on the accomplishments of 2014 and the Nobel. But it seems like there was a lot of fascinating work that went into it prior. But so much has kind of just come together over the last few decades. It seems like a lot has kind of all. There's a lot of things that were uncovered, but a lot of things have been complementary to where once one thing is grasped, it unlocks a whole new field, and it just kind of snowballs at times.
Yeah, it's true. It's true. It was the whole field was moving forward over the decades because it's not exactly trivial to understanding different brain areas and recording from them, and really our ability to do it over the last even couple of decades improved drastically in comparison. Just to give you an example, if you look at technology in the early 70s that John O'Keefe used to record from these cells, special properties of the cells, it was literally just a camera hanging on the roof, on the ceiling. And he would press himself manually, trigger the camera to take a picture every time he would hear a cell from amplifier. So from that, moving to what we have now with all the cameras, high-resolution cameras, automatic recordings, silicon probes with thousands and thousands of channels. When you compare that to, having 3 channels and recording, triggering with your hand the camera, it's massive improvements, and that's why we see more integrative research at the moment. That we start looking at not only response of specific cells, but actually how these cells interact to provide us with this cognitive map.
Interests me too that even in Dr. O'Keefe's progress, that one of the elements, aside from technology itself growing, that even common things like video games got utilized along the way. That I thought that was hilarious. That Duke Nukem and I think another game by Sony, like London Taxi Driver, that those were 2 tools that got utilized in being able to really hone in on the hippocampus as kind of this central location for cognitive mapping.
It's true. So his lab was one of the first one which started actually hacking these games to provide, because really they needed comparison between human and between animals, because on animals our best research, or at least the research we best understand, is done on spatial properties. And if you look how a fMRI machine looks like, of course you can't put on the human head and let him walk or her walk with it. So they had to make something that people could navigate without moving their heads. And as I said, it was really great that all these gaming engines were starting up at that time.
When we're taking a look at the basics of the ingredients we need for a cognitive map, there's a number of key cells that have already been found. You'd mentioned place cells. It's just something that will, as human or as animal moves around, it gives a localized location that's understood. In complementing that, there's also grid cells, which can give a sense of direction, kind of almost a north, south, east, west type approach.
Grid cells, in a way, I would say they're a bit similar in places, but because of their periodic properties, you maybe can look at it as a way to relate places. Not as imagine the place from a response of place cells, and grid cells let you translate your perception when you move through space. So, for example, you moved 3 steps, and many grid fields turned up and fired, and that's how they signal to hippocampus, to place cells, that different ensemble of place cells should start activating.
Okay, and then beyond that, we do have head directional cells that aren't as much concerned with movement as much as it is the direction of the animal or human's head at the time.
Yes.
Yeah. Okay.
Yes.
And then beyond that, we get to a real interesting facet of boundary cells.
Boundary cells, you probably already saw when you did your research that they fire only at the boundary, and typically only at one of the boundaries, let's say only at the north boundary. And if you put some sort of object inside the environment, it would fire at the object at the south part of the object where the north boundary would be, and at the north boundary. So, but we still working on understanding mechanistically how they're connecting to each other, and whether some of them are just a result of calculation of more basic cells.
Yeah, this is where it starts to get super interesting, because you start talking about the whole fact that deep within our brains, or within an animal's brain, they start to construct literally a map of the environment that they're inside. Cells will start to play a specific role, and you start getting to this uncomfortable moment of like, well, gosh, that means that every animal is actually doing some basic geometry about the space they're inside of, just as much as we are. Which is, which you start getting to this complex thing of like, well, wait, so is geometry something innate that we have got, that we've used all the time? Is it something that goes through all mammalian structures? I mean, there's some really big questions that start to pop up out of this.
Yeah, it's true. And this is one of the aspects we were looking at, how geometry affects the animals, how actually the perception of space is what's happening to it. So we are still working to understand that. So what we showed already, that these, for example, grid cells, which are these beautiful periodic cells in the square or circular environments, if you put animal in the distorted geometry environment, these cells just become non-symmetric anymore. And the idea is that they have to get some perception of space, that they have to understand that this geometry is not a square, for example, but what does it mean for animal? We are still working to understand. So currently we're doing experiments to see whether they can't measure space, measure distance in that area anymore, or it's just distorted in a way like you could stretch. Imagine you just push together walls, and instead of square you make trapezoid.
Okay.
And maybe, animal would still use calculations that it's a square, but make errors because of this squeezing. So we're still working on psychological understanding what's happening.
But even when we do that innately, just almost immediately upon examining where we're located, we might have this rough map and rough set of boundaries that pop up, but as we move around in it, we start to firm it up. And it starts to become refined. Okay. Yeah, that was one thing in your, you did a great article in the Journal of Physiology. You had stated that for some room shapes, we can adjust, or animal adjusts, really fast. I mean, a matter of a day or something. But in other shapes, it can be a whole lot longer.
Yeah, so we don't know exactly why, but the more complex, the more complicated geometry is, the more it seemed for animal to take, as you said, actually for these cells to become fine. So we don't know exactly whether animals' perception is following the development of these cells, and you're looking at it. But yes, the more complex environment, the more time grid cells need to become more symmetric.
It's amazing. So, the one for us is, after we've read your work and we've kind of seen developments that have happened over the last couple of decades, for us it really starts to make some big implications toward maps in general. And for the first time in my life, I think I looked at a map and I thought, gosh, this isn't necessarily some socially evolved, process of how we would chart the world, as much as it's almost kind of a paper receipt for how our brains work.
That is an interesting idea, especially keeping in mind that we have neural representation of space literally in our brain, almost maps. But it's difficult to say with human, because with human we know that many of these concepts do transfer, that we already can record from grid cells or from or place cells in human, and we can see that many of them transfer, but we don't know exactly how this works, because human, for example, compared to rodents, are much more visual creatures. So we use our vision far more than them. In the rats and mice, vision is not really good, to be fair, but for us it's we estimate things with our vision much more than they are, and it's difficult to say how does it affect perception of geometry.
That comes up to an interesting subject. I made my own terrible, simple analogy of saying there are kind of two styles of maps that we see. If we were to make some terrible generalization, we could say that there's kind of maps that are pictorial and that are more route-based, where you're going from one landmark or kind of point of interest to another, yes, and you just kind of tie those together as ever you can. And then there's more data-driven, more objective cartography that, will base on a grid, will allow you to improvise moving around a whole lot easier, and is something that tends to be, for a lot of people, tends to be a lot more dry, because there's no real sense of how that place is understood or utilized or what it means. Mm-hmm. Mm-hmm. But I hate to say it, the more, and perhaps I'm in terrible error just by, anthropomorphizing so much, but it really reminded me of the same kind of situations that happen deep within our understanding of GPSing where we're at, is that there's a structure where you'll navigate by route. There are other structure where you will navigate just by kind of a grid and by other relations. And that's kind of true of how just innately how we're wired. Is that correct?
It actually is correct. So there are two ways, broadly there are two ways of navigating. One of them is forming this map of the environment, and then you can, I'm talking about animals now, so you can map environment and create shortcuts or just find a correct way without just calculate the correct way where animals should go, even without animals seeing the goal. And other way is where animal would just have beacon and go towards the beacon or several beacons and have this, as you said, route with some sort of big cues. And the interesting thing is that it seems that for this kind of navigation, for beacons, hippocampus is not really necessary, that most of the work of following the beacon is done outside the hippocampus.
Is that entorhinal, or where's that located?
No, so we don't know exactly where it is located. I mean, it's distributed. Okay. But entorhinal cortex is the whole. Is part of what we call hippocampal formation. So it's, most of these special areas are pulled together under hippocampus name.
Okay. I suppose that was one thing that was radical for me too. Is the more I've listened and studied and tried to pay attention, it seems like we went from a point in time back in the 19th century where this little chunk of your brain is going to take care of hunger. This part of your brain will take care of love and other things. There was this basic phrenology approach to where this chunk does that, this chunk does the other. Well, at the end of 19th century and into the early 20th, we start to realize it's like, oh no, it's a lot more complex than that. There's layers and structures, and we don't understand how they feed into each other, and it becomes a lot more complex. Well, with this study, it almost gets us back to an uncomfortable point of, oh gosh, it looks like there are specific points that do the particular heavy lifting of one function.
So we do have this idea. So it's not exactly that we have this one area and on its own it would do everything, but we have areas with crucial nodes, so to say, where a lot of information are merging from different areas. For example, if you want navigation in hippocampus, we have to get input from sensory areas, for example, from vision, visual flow, to see how we're moving, from locomotion, from motor areas, from response from our muscle. So basically, we have to get all this information pulled, calculated, and heavy lifting may be done in hippocampal formation, where we also, again, we have many different substructures, and then moved forward to wherever it's necessary. Navigation is mainly done, I would say, in hippocampus, which related to what we would call cognitive map. But really, the whole brain is a big hub where you can't separate one part from another very neatly.
Yeah, there's a great series of talks that was done. He basically starts out the whole thing by saying, look, if there's a problem with, say, tornadoes or storms, you don't get into the subject and try and quote unquote figure it out by, all right, let's break down these clouds and figure out exactly what's inside them, and then we'll be able to unleash and understand it. it doesn't work that way. You don't break down a cloud into, oh, there's humidity and some movement, and all of a sudden you understand everything. There's so many scalar levels of change that happen that it's not that simple.
It is. So now we are actually getting tools where we can do approximation of that. So, for example, optogenetics, we can literally turn off a particular cells or particular area in the brain and see what's happening. But again, the issue is that everything is so much connected that even if you turn off, let's say, clouds in that metaphor, you don't know, maybe some other part starts compensating. So you need to do a lot of checks, and we're already getting the position where we can look at that more closely and even at compensation. But it's true. It's when it's so complicated, so interconnected, it's difficult to disentangle.
While obviously mammalian brains, there's going to be a lot of variety, and structurally there's going to be some basic undercurrents throughout. They're going to be consistent. But as you go from mice to humans to dolphins to a million things in between, does it seem like structurally there are these unifying kind of momentums of how cognitive mapping happens?
So it certainly seems that there are a lot of commonalities. So if you look at in general in the brain between the mouse and rat, it's bigger by almost factor of 10, and the basic areas are the same. If you look go from rat to human, you will see that certain areas like prefrontal cortex will be far bigger in human, but other areas will be similar with similar layers, similar cell types. In many ways, they have a lot of similarities, and we already know that even functionally, many of the cells are acting quite similarly.
I'd be so curious to see, for species where I expect they don't have as many bounded environments, whether that's, dolphins or whatever. I'd be curious as to how structures reform or how it gets reapplied. Or if there is any application, if dolphins end up swimming, in some limitless or near limitless context, how does it all make sense again?
I don't think there is such research with dolphins, but what we do know is what happens with bats. Bats are also, they can fly for tens of miles, and they have feeding grounds, they have sleeping nests, and we know that actually they can encode this whole environment within one big spatial structure. So they'll have these massive place fields on the way, and they still, we don't know the limits of this. We know better the limits, for example, in rats, that if the room becomes too big or a rat has to walk for too far, the whole representation will create a new map, a new representation. So you have, for every room, you have some different representation, different placeouts. So we know that they can't be continuous for really long corridors, let's say, or really long, big areas. But for immediate surrounding, it's quite continuous.
Okay. I was interested too. I had Dr. Moser has done some really good overviews and some fascinating work as well, he and his wife. And he had pointed out that there's a scalar nature to the cells themselves and how they will work at one level and then will upgrade at 1.42, will scale up again, and then at that same ratio can scale up again. That there was almost a weird golden ratio of how we enlarge maps of areas.
Yes, this is particularly for grid cells, because in grid cells we have grid cells can be described by mainly one parameter, which is how far away are fields within the grid cell. So in grid cells we have many different fields with the same distance from each other, and if you go from a different part of entorhinal cortex where they're found, you will find that, as you mentioned, that they'll become bigger and then bigger. And golden ratio, so far the best models, I would say, at least the most convincing models, are the ones which deal of uniqueness of place. So at a certain ratio of grid cells, you can uniquely encode much bigger place. So, for example, imagine you have a grid cell of 1 meter and grid cell of 2 meters. And every 2 meters, they will completely overlap. So they're really bad at encoding large environments. But if they are meter and 1.5 meter, they will overlap only at 3 meters. So you see environment's already 50% bigger. And it seems that, what was proposed, that this ratio, which varies from 1.2 to 1.5, depending on the experiments. It's optimal for actually maximizing the environment you can have computationally.
I have to say, it gets me back to some jaw-dropping moments of seeing the intricacies and kind of the necessary interrelated complexities that happen. And it kind of puts you in a state of awe at some juncture. The more you kind of drill down into it, what can feel so innate and natural, when you back off and really start to understand the subtleties of it, it's amazing. And that's not even big enough. It's stunning and almost unnerving at times to kind of look at how many fine kind of gears have to work together for things to function like this.
No, it's true. It's this whole beauty of these structures actually what mainly made me move to neuroscience.
Dr. Bauza, I cannot thank you enough. This has answered questions and has, once again, it's kind of got me excited. I don't know what it is about the topic, but it seems like there's some really fascinating stuff going on from many different vantage points. So, man, thank you.
No, you're welcome. It's really exciting for me too. So it's always happy to share that excitement with other people.
Awesome.